All right, everyone, we are going to go over something called the hypersensitivities in the body. Now, we can define hypersensitivities essentially by calling them too much immunity. Too much immunity. What it is going to mirror, essentially, in a deleterious way, is the typical immune response that the body wants to engage in and needs to engage in to remove a pathogen or an infection. However, it's going to typically do those things in a negative way that would affect the body. So just to review, when we are talking about the immune response, we are starting at the point where some sort of pathogen is entering the body, a foreign pathogen, an antigen. It is engulfed by a cell that we refer to as an antigen-presenting cell. And remember, that presenting term comes from the fact that once the antigen is brought in and processed, a portion of that antigen will be displayed on the surface of that antigen-presenting cell. And that antigen-presenting cell, displaying that antigen fragment, remember, everything in the body is going to be attracted to one another by shape. So the specific antigen, when processed, will have some sort of surface characteristic that's specific to it. And that is called an epitope region of the antigen. By virtue of the circulation, chances are that eventually a CD4, or what we call a T helper cell, will make it to that area and have displayed on its surface a T cell receptor, which you see here in bluish purple, which matches the epitope region that's displayed by the antigen presenting cell. Those two things engage in a binding interaction, and in response to that, we have the release of cytokines like interleukin 1, interleukin 2, and those are released by the antigen-presenting cell and the T helper cell. When those cytokines are released, so now we're here, the T helper cells, we have these cytokines, these cellular messages that are soluble and released into the bloodstream. We have the activation of two concurrent arms of the immune system. We have the cell-mediated, which is going to involve T cells, and we have the humoral side, which is going to involve B cells. So in this picture, we can see that the B cells are associated with antibodies. Remember that when activated by the cytokines released by the T helper cell, B cells differentiate into memory B cells, which are going to display the antibodies with specific matching regions on them, similar to the epitope region of the antigen fragment. So that's our memory B cell. So that is hanging out for decades at a time in the circulation for the chance that we will have contact with that specific antigen again. And that is the primary basis by which vaccines work. We come into contact, in this case, with a controlled pathogen. The body engages in this response, makes these memory cells with antibodies on them that match the fragments of the pathogen. The other thing that happens when B cells are stimulated by cytokines is that they split off into plasma cells. And the plasma cells are the ones that are the antibody-making factories. So they are actively secreting antibodies, again, which have a receptor site on them that match the epitope region for the specific pathogen. So while the B cell arm is engaged in making memory B cells and plasma cells, the CD8 cell-mediated side, these killer T cells we call them, are going to be activated to come to that area and start killing or engulfing cells that are tagged for destruction. And one of the things that we'll go over today is really any cell that is tagged with an antibody is essentially tagged for destruction in the body. So a killer T cell sees no problem killing a cell tagged with an antibody. And killer T cells secrete cytotoxic substances to degrade a cell, which is different than a plasma cell making antibody, which would just attach to a cell. Okay, so this immune response is a normal immune response, it's a healthy immune response, and it exists to rid the body of a pathogen. When these things happen against our own healthy cells, we refer to them as hypersensitivities. Here is a table going over each of the 4 types of hypersensitivities, which I'll explain to you in this lecture. And in this table, you have the different hypersensitivity types. So we have type 1, 2, 3, and 4. They are going to involve either an immunoglobulin, like types 1 through 3. They involve different antibody or immunoglobulin types, whereas type 4 is going to involve T cells. There are specific antigens that are correlated with each of these immunoglobulin reactions. Here it has the mechanism of activation for each of these hypersensitivities and then examples of the hypersensitivities. So we'll go on and describe this functionality. So we'll start with probably the most well-known of the hypersensitivities, the type 1 hypersensitivity, which is an immediate hypersensitivity and is referred to as anaphylaxis. This is also just referred to as an allergic response or an allergic reaction. And this type 1 hypersensitivity is mainly going to involve the IgE antibody, the IgE immunoglobulin. And if you're familiar at all with allergies, you'll know that allergies are generally associated with the IgE antibody. So if you wanted to see if somebody had a particular allergy, like a peanut allergy, for example, you would test for the existence of an IgE antibody with a fragment on it that would bind to or recognize peanut as an antigen. So the key here is that peanut is not a typical antigen, right? If you eat a peanut, it shouldn't kill you. The body should not recognize that as an antigen. But in a hypersensitivity, the body does recognize that as something that is a direct threat to the physiology and needs to be removed. Developmental biologists have agreed that these IgE antibodies seem to have been developed specifically to detect and remove the body of parasitic worms. There aren't so many parasitic worms just out generally, given changes to drinking water and sanitation. So really, these IgE antibodies function in a modern society with our modern biology to be triggered by allergens, to give an allergic response, those things that are ranging from either just annoying and seasonal to life-threatening in some cases. So what you see in this picture is our first introduction to what a type 1 hypersensitivity looks like. And in this case, the antigen you see is pollen. So pollen from a plant, a flower, or even from honey made by bees, which carry with them some fragments of pollen from the flowers. Fun fact, that's why they tell you to buy local honey to help with allergies. You're becoming essentially used to fragments of pollen or other plants by ingesting honey and then having a kind of undercover immune response to them to make you more able to withstand secondary exposures to them. So anyways, we have the pollen, which is recognized by this body as a pathogen that threatens the life of that person, of whatever organism this is. So the allergen binds to a specific portion on an immune cell. In this case, we're looking at a B cell. Remember that 3 hypersensitivities, 1, 2, 3, 4, all involve immunoglobulins, and an immunoglobulin is associated with the B cell or humoral immune system. So the pollen binds, which means that we're going to have antibody, which recognize a portion of that pollen fragment, that epitope portion, to be created. So now there will be antibodies, immunoglobulins in the body that are able to recognize that pollen. And once those antibodies are created by the plasma cells, they need to go somewhere. They're not just going to hang out in the bloodstream and wait for destruction. Your body doesn't readily destroy anything that it's worked so hard on. So where those immunoglobulins go, so these ones that are specific, in this case to pollen fragments, they bind to the FC, the crystallizable fragment portion of mast cells. And that's called sensitizing a mast cell. It's essentially priming them. So the mast cell sits there now, not just with histamine inside of it, but also with antibody bound to it that are looking for pollen. They're ready to bind pollen. So when there's a secondary exposure to pollen, and the pollen binds to the Fab portion of this antibody, we get degranulation of this mast cell. Degranulation means that it removes its granules, and in this case, its granules are filled with histamine. So the binding of this pathogen to those antibody fragments induces this degranulation. And you might already know that histamine is associated with allergies. Histamine causes a reaction. It actually drives the full inflammatory response. It causes these endothelial cells to contract and help with the permeability increase that we see in the inflammatory response. So this is another picture that we see in this example with an antigen-presenting cell with the pollen antigen. We've just added the earlier steps to this process with the T helper type 2 cells. Those are the ones that release specific interleukins, specifically 4 and 5, which activate B cells to engage in the plasma cell process whereby they make IgE antibodies against that particular pathogen. Again, you see that these antibodies, they go here, they sit or prime the mast cell so that when we have the secondary exposure of the pollen, we have degranulation of this mast cell and a large release of histamine. Another thing besides just the antibody sitting, priming the mast cell, is that you'll see these antibodies cross-link, kind of like they're holding hands. And what they're doing there is joining their forces together so that when the pollen binds that second time, you get an even more dramatic reaction, even more release of histamine, causing this anaphylactic reaction. So the first time that there is an exposure to this pathogen, this pollen, all of this stuff leading up to the creation of this IgE antibody, you wouldn't even notice it. It's under the surface. Your body is doing this without your knowledge. And now the mast cells are primed with these IgE antibodies, these immunoglobulins that are ready and able to bind that specific pathogen and have a dramatic response referred to as an allergy. So this cross-linking and this priming or sensitizing of the mast cells is what drives this dramatic reaction you see upon a secondary exposure. This is another picture showing you the general process of this immediate reaction. Here in this lighter portion, we see the antigen-presenting cell processing that antigen fragment, displaying it to T helper cells. The T helper type 2 cells are the type by releasing interleukin 4, interleukin 5 to call the B cells to the area so they differentiate into memory and plasma cells, the creation of IgE antibodies, and the binding of those IgE antibodies to these FC receptor sites on a mast cell, which is filled with histamine. So now the cell is primed, it's sensitized, it's ready to wreak havoc and release all of its histamine. And again, whenever histamine is released, we see that we get dilation of those precapillary sphincters, we get passive blood flow into the capillary beds because of that. That's going to increase permeability in the cell walls. And then that increased permeability allows for more space to let the white blood cells out. Again, this would be a good thing. We want the white blood cells to come out in an inflammatory reaction. Again, the problem here is that this is a dramatic reaction to something that is not a true threat to the body, like pollen in this example. So because we have these, again, these cross-link antibodies and these primed sensitized mast cells, the degranulation, the release of this histamine is really dramatic, and we get this very pronounced secondary response. Okay, when you have this immediate hypersensitivity, so binding of antigen to IgE antibodies on mast cells. Here again, we see these cross-linked IgE antibodies binding that pathogen. The mediators in this case are going to be mainly histamine. We'll talk more about the other factors later. Those involve things like serotonin, like complement, heparin, which is going to be involved in blood clotting. We'll also see the formation through the destruction of certain cell types and their cell walls, which are made of phospholipids degraded into these pro-inflammatory proteins. So these prostaglandins, PGEs, and these leukotrienes, LTKEs, which you may know are blocked by a variety of drugs like NSAIDs and leukotriene inhibitors. So when things like these prostaglandins or histamine, serotonin, other chemically mediating substances are released into the circulation, we have these typical reactions you would expect in the inflammatory response. So the vasodilation, increased permeability, smooth muscle spasm, meaning that it initially is going to cause contraction of the muscles surrounding small vasculature, and then eventually dilation and relaxation of that muscle so that there is a widening of those spaces between endothelial cells to allow for chemotaxis and then the migration of these white blood cells out of the circulation. So those are the things that we would expect. However, in this immediate hypersensitivity, again, we have this dramatic secondary response, this immediate anaphylaxis. So there's two types of anaphylaxis. There is local, that's also referred to as atopy or atopic. So if you've heard of atopic eczema, that's referring to local eczema. So it's relegated to the region generally of exposure to this allergen, which is different than systemic anaphylaxis, which is going to involve the entire body. In local anaphylaxis, that is generally going to occur in response to contact of the pathogen with either a portion of the epidermis or to a mucosal membrane. So if we're thinking about pollen, for example, that would enter the nose, the nasal cavity, and then on that mucosal membrane inside of the nasal cavity, enter the bloodstream and then cause this response. So its initial contact is going to be at the mucosal barrier or epidermis. Some people get itchy from pollen on their skin, or you might be, I'm allergic to scented detergent. So that's contact with those things on the epidermis. So again, these local anaphylactic reactions, they're usually exposure at the mucosal surface, epidermal surface, and some examples of local atopic responses are things like hay fever, which we call allergic rhinitis, inflammation of the nose, essentially. Think of a rhino and its big horn on its nose. So rhinitis, inflammation of the nose area. Urticaria or hives, angioedema, some forms of asthma. Conjunctivitis is inflammation of the conjunctiva on the eyes, and again, that atopic eczema. So this is different than systemic anaphylaxis, which generally follows injection of an antigen, but in some rare cases, it can involve contact of that allergen with the mucosal membrane. And that, an example of that would be the peanut allergy. So that would be an ingested peanut, the peanut making contact with the surface inside of the mouth, inside of the esophagus, the larynx, and that would cause this traumatic reaction. But typically you would see this with injection. And another example of an injection, besides like through a syringe, would be with a bee sting. That bee is injecting its nasty stuff into you, and that's why they're awful, and we should all hate them, except for, the flowers and trees that they keep alive, I guess. So when the entire system responds, that's not just the immediate area that is touched by that pathogen, by that antigen. It's going to cause a body-wide response to something. So that is when you might see all over hives and itching and redness, possible respiratory distress, so wheezing, difficulty breathing, shortness of breath, laryngeal edema, which is swelling around the throat area, difficulty swallowing. You can see GI symptoms like vomiting, cramps, diarrhea. Laryngeal obstruction is when you really can't swallow. You have difficulty getting any air in, and you have to have different medical interventions then. Dysrhythmias, dys means bad or deranged, so that would be an abnormal rhythm. And because histamine is a vasodilator, you can see anaphylactic shock, which is shock as a result of the release of too much of a vasodilating substance. And shock at its very core is really just not enough blood getting to the organs. That's it. So if that shock is severe enough and organs start to suffer ischemic damage due to loss of blood flow, that can be deadly in some cases. So all of these things can be reversed at the local level, definitely by an antihistamine, because this response is driven by the release of histamine. For more systemic things, it's always going to be most important to treat anything respiratory-wise first, because if you can't breathe, you can't live. We'll learn more about this later, but epinephrine, because of the receptors that it binds to, it causes contraction in so many places, like the heart rate goes up. However, in the bronchi, it causes dilation. So you get bronchorelaxation rather than bronchospasm, which encourages breathing, and breathing encourages survival. So that's our type 1 immediate hypersensitivity. Just to point out, we call that anaphylactic reaction or anaphylaxis. Whenever you see oid at the end of something, that means kind of like it, but not quite. So we have an anaphylactoid reaction, which resembles an anaphylactic reaction, but isn't one completely. So you see these legs, you can tell that there's something wrong with them. It looks like there's a rash around where the socks or stockings were, so there's some hives. There's some darker purple areas, which we call purpura. Purpura, they almost look like bruising, but what's different about a bruise and purpura doesn't blanch, meaning if I took my finger and I pressed it as hard as I could into this purpura, it wouldn't change color. If right now you took your finger, and I'm doing it right now, you can't see me, but I am. So I'm going to press my finger as hard as I can into my forearm. And if you're doing that too, when you take your finger away, you see that within a second or two, the skin goes from a blanched white color back to normal color. And that's looking at capillary refill time, but that initial blanching, that turning white, that is something that you would see in bruises and in typical tissue, but it is the way that you can tell something is purpura. It does not blanch. So getting back to this anaphylactoid reaction, it resembles anaphylaxis in that it looks the same. However, it's not caused by this release of histamine. It's not caused by these IgE immunoglobulins. What it can be caused by are just bizarre things. An example is dark meat fish. So for example, let's say somebody was out ice fishing or fishing in general, like some fish are white on the inside and some fish are dark. When dark meat of fish is not properly refrigerated, it contains a substance called histidine. And histidine over time will be converted into histamine if that meat is not refrigerated. So I mentioned things like ice fishing or fishing if you're outside and you just, gut that fish and keep the fish meat out and want to have it later. The danger is that the histidine has been converted into histamine. So when you eat that fish, you have this anaphylactoid reaction. It's not that that person is allergic to fish. The problem is that they just consumed a large amount of histamine because of that conversion process. So again, that's called anaphylactoid. It's not the same as anaphylaxis, as type 1, but it resembles it. And the next shows you how to tell the difference between them. So an anaphylactic or anaphylactoid reaction. So the first question you could ask is sensitization required? Meaning, is an antibody, an IgE antibody bound to a mast cell required? Do you have to have a first exposure and then a secondary exposure? For anaphylaxis, yes, that's the whole point. That's the mechanism by which they work. For anaphylactoid, no, it would be caused by something like eating improperly refrigerated fish. Can a reaction occur on first exposure? For anaphylactic reactions, no, it can't, because the body has to make those IgE antibodies, which then have to bind to mast cells and cross-link with one another to be ready to bind that pathogen for this reaction. For anaphylactoid, sure, first time you eat improperly refrigerated fish, you can have this reaction. We don't have any of this immunoglobulin activity. Next, how much exposure is needed to elicit the reaction? For anaphylactic, very little, right? You come into contact with a pollen fragment with, like some people, if they are allergic to peanuts and some touches their plate, for example, they can have a dramatic reaction. Where anaphylactoid, if it's the fish, for example, they have to eat a lot of that fish and then they have that reaction. They don't just touch the fish and then have breathing difficulties. And then the most important predictor, really, from a clinical perspective, is it predicted by an IgE allergy test? So if you bring them to the lab and get testing done, you can detect an IgE antibody for various allergies. There is no IgE antibody for anaphylactoid, so it cannot be predicted or found in any way by an allergy test. So that was the longest one. That was our type 1 hypersensitivity. Moving on, the type 2 hypersensitivity, I'm sorry, these words are all messed up. Sorry about that, is called antibody-dependent cytotoxicity. So toxicity to cells. This is a way that the body typically acts, remembering that any cell in the body that is covered in antibody is tagged for destruction. And the whole point of that process is that that cell, which now has antibody attached to it, in whatever iteration, would be an infected, bad cell. So that when something like a natural killer cell comes along and recognizes the fact that there's an antibody there, it knows that this cell is bad and it's going to release stuff to kill it. Now it's dead. It's got X eyes. When this happens to normal self cells, so in this case, this would be a normal self cell. Look how happy it is. It's so happy. For whatever reason, in autoimmune diseases, the body makes antibody against healthy cells. So the body's like, ha, red blood cells, or ha, thyroid, I hate you. I'm making evil antibodies. They're going to tag you. And once you're tagged, you're going to be destroyed. And that's exactly what happens in type 2. It is the normal function of the system. It's the creation of antibodies, but in this case, they're rogue, bad, evil antibodies. Look at that evil smile that recognize self cells. So see here where it says self antigen, the body created these evil antibodies that are going to recognize and then bind to this self antigen portion of the cell. And once that antibody is there, this nice happy cell is now tagged for death. It's tagged for destruction. So the natural killer cells, see, he's got a straight face. So he's just doing what he's told. He knows that when he's called to that area, it needs to recognize any cell that has antibody on it as a predator, as an abnormal infected body cell. So it just does what it's supposed to. It recognizes that and it engages in this non-phagocytic cytotoxic action. So some cells, like natural killer cells a lot of the time, release cytotoxic materials. It also can activate the complement cascade. And remember, complement cascades are many steps long, but they involve the formation of something called the membrane attack complex, which is the creation of these cylindrical structures. So they're just tubes from above, from the side, they look like this, and that tube is inserted into the cell membrane. That's called opsonization. And once there's a hole punched in the cell membrane, that cell is going to lose all the stuff inside of it, and now it's going to die. So this nice, happy body cell is now a dead body cell because, for whatever reason, the body made antibody that recognized that cell as bad. And this is a basis for a ton of autoimmune disorders. Again, that's type 2 hypersensitivity. This is another illustration of type 2 hypersensitivities. The B cells activate plasma cells, which make antibody, but they make antibody against healthy body cells. Like here is a red blood cell, and the body's made antibody against it, which will tag the red blood cell, and now it will be destroyed. We can also see this with receptors that are destroyed. There's a disorder called myasthenia gravis, where there are antibodies that are created that recognize specific receptors. And with myasthenia gravis, it's acetylcholine, ACH, and acetylcholine is associated with muscle contraction, and you get the destruction of these receptors because they are tagged with antibody. That means the ligand or ligand, whatever you want to call it, ligand or ligand is a term for anything that binds to a receptor. It could be a drug, it could be a neurotransmitter. In my example, it would be acetylcholine. The acetylcholine would love to bind to this receptor, but it cannot because, first of all, there's antibody on it, and then second of all, it's going to have a destroyed receptor because there's antibody on it. Again, we call this this associated cytotoxicity. This is driven again by these immunoglobulins that are created and tagging healthy, normal body cells. Next, we have type 3 hypersensitivities, which is a very adequate name. I like it a lot. I like things that are named in a way that describes them. Type 3 is called immune complex mediated hypersensitivity, which means that it is a process that's helped along by the creation of these things called immune complexes, which essentially are collections. So in the dots here in my picture are antigen, and then we have antibodies binding to that to create these little complexes. And the key here in type 3 is that in a healthy immune response, the amount of antibody that's created, so the amount of antibody that's created in response to the antigen is a good ratio. It's matched, so like a 1-to-1 ratio, which means all that antibody that's created, it's an actual substance, so it's going to travel in the bloodstream, connected to these different pathogenic substances. And then eventually it'll be neutralized by the spleen after it's metabolized by the liver. And those things are big, so it's easy for these big or large amounts of antigen antibody substance to move through the bloodstream and be neutralized and then removed. The problem here is when there is a really dramatic overproduction of antibody. So you'll see this in cases of strep throat in kids where there's an overreaction to the pathogen. So there's a lot of antibody made, and when there's a poor match, you end up, as a side effect of that, with some little antigen antibody complexes. And they are so small that it's really hard for them to be carried away by the bloodstream. What that means is that they can get lodged in the microcirculature. And here's the key. If they get lodged, they're in vessels. And remember, if they get stuck there, there's still antibody. And what happens to any cell tagged with antibody? The answer is that it's tagged for destruction. So even though this is a healthy endothelial cell inside of a vessel, this neutrophil sees that there's antibody there, thinks that it's helping, and then eats away to remove this, but it also is going to remove a portion of the endothelial cells. And we'll learn more about this later. If you've already learned about clotting, remember that any time the basement membrane is exposed, that's going to activate the coagulation cascade, and then you'll see thrombi, in this case it's microthrombi, very small blood clots form in that area and damage the vessels. Also, there's big holes that are going to be created in these vessel walls, making capillaries really leaky. You see this driving a lot of glomerular disorders where protein is allowed to leave the vessel walls because these holes now are too big and allow for too much permeability. All right, here's another picture of this type 3 immune complex, mediated hypersensitivity. Here's our immune complex deposited in the vessel wall because it's lodged there. The antibodies in general is going to attract these specialized white blood cells who know how to recognize antibody and know to kill anything that it is tagged to, and that creates this destruction that you see. You see this a lot of the time in the kidneys because of the fact that a lot of blood flows there. They're leaky vessels to begin with, and then you can see protein moving through the glomerular walls, which should never happen. Okay, finally we have type 4, which is called cell-mediated delayed type hypersensitivity. So first of all, it's called cell-mediated because it involves T helper cells, not an immunoglobulin. And the second part, the delayed type, means that the reaction doesn't occur immediately. It actually peaks within a day or two. So exudate formation is about 38 to 72 hours later. And this is in response to persistent types of antigens. You see this with really sticky substances like poison oak or poison ivy, with organ grafts. So if somebody gets a donor organ, that's persistent and it's not going away. And then you also see this if you ever had a TB test and you've had tuberculin injected underneath your skin. You wait for that to swell up, and it takes, a day or two for that to happen. And what you're looking at is a live reaction in a type 4 hypersensitivity. So this process is going to involve the T helper cells activating the immune response, but there is an antigen that's persistent that won't go away. So it's essentially this reaction happening over and over and over again and happening a lot. So you don't just have a little bit of activation of this, you have a continued activation of it. It's dramatic, and you get this extreme reaction causing lots of exudate development and a more persistent reaction that takes a while to pop up. Okay, and here is another slide showing you the mechanisms and the descriptions for each of the types of hypersensitivities. Make sure in my class you know the different types of hypersensitivities, what activates them, and some examples of disorders with those hypersensitivities. All right, thanks. Bye. Or if you